The innate immune system has two lines of defense: innate immunity and adaptive immunity. The focus of this study, inflammation, takes place within the first immunological defense, innate immunity. Macrophages are well known as effector cells, which respond to stimulus and are part of the innate immune system. Macrophages are present in almost all tissues of the body. This allows them to rapidly secrete a large range of cytokines that can influence an inflammatory response. There are two phenotypes of macrophages, pro-inflammatory M1 and anti-inflammatory M2, which both secrete specific cytokines. Naringenin is a flavonoid that can attenuate the production of pro-inflammatory cytokines. It is also found to have a broad range of effects on human health. It promotes carbohydrate metabolism, increases antioxidant defenses, and modulates immune system activity. The purpose of modulating the M1-M2 balance is aiming to avoid the dysregulation of the immune pathway. Dysregulations can lead to serious disease such as autoimmune neuropathies, arthritis, and cancer. For this study, the focus was on naringenin's ability to reduce the production of the pro-inflammatory cytokine interleukin-12, and to also measure the IL-10 present after naringenin treatment. I first attempted to extract naringenin from grapefruit peels using SFE, supercritical fluid extraction, with a co-solvent of reagent alcohol and a main solvent of CO2. To observe the cytotoxicity of the naringenin standard I used, I first performed an MTT assay. I started by growing THP1 monocytes, which then I differentiated into macrophages using PMA. I then treated the macrophages using 5 different concentrations, all completed in triplicate, on one group only receiving naringenin treatment and the other group receiving naringenin treatment with a co-stimulation with LPS. The percent cell viability was then determined. After determining the cytotoxicity profile, I then treated the macrophages with a high and low dose of naringenin, again on the treatment-only group and the co-stimulated LPS group. Following a 24-hour incubation with the treatment, the RNA was extracted from the cells and the cDNA was synthesized and RT-qPCR was performed. The results of the MTT assay were analyzed using a one-way ANOVA to produce the percent cell viability. The treatments were normalized to the methanol vehicle control, which was 3%, and there was no significance in any of the data. The two treatments that were chosen were 0.04 millimolar and 0.1 millimolar. A two-way ANOVA was done on both differentiated macrophages treated with naringenin and the LPS co-stimulated macrophages to determine the change of IL-12 expression. The IL-12 target gene data was normalized to the housekeeping gene GAPDH. The results of the RT-qPCR showed that naringenin did decrease the amount of IL-12 present in both the treated macrophages and the co-stimulated macrophages. The results also showed that there was a trend of naringenin reducing the amount of IL-12 present, while no significance was found in the data. Naringenin was not successfully extracted from the grapefruit peels using SFE. Stimulated M1 macrophages that were treated with naringenin did show a decrease of the pro-inflammatory secretion, and there was no secretion of the M2 phenotype cytokine IL-10. Future directions include finding different methods of extracting naringenin from grapefruit peels. This could be extracting from only the albedo of the peel instead of the whole peel and attempting to extract from peels that have been dried. Investigating the effects naringenin has on anti-inflammatory cytokines that are more abundant than the IL-10 cytokine to determine if naringenin treatment does induce the expression of M2 phenotype cytokines. I would like to express my gratitude and appreciation to Crystal McFadyen, Jessica Livingston, Marla McKenzie, Michael Gibson, and Alea Quilty for their encouragement and assistance during research and throughout the bioscience program.